Trinity Cultural Academy and NEPA's New India Publishing Agency welcomes you all for the presentation on Tidbits 5 Dormancy Breaking Treatment. And this topic comes under the subject plant science, that is seed science and technology. This material is prepared by Dr. K. Vanangamudi, formerly Dean Agriculture, TNPSC Coimbatore, and so on. Now let us enter into the subject. In the previous video, we have seen that what is dormancy and why dormancy is needed, what is the physiology of dormancy and the classification of dormancy and its causes. And in this video, we are going to see about the methods to break dormancy. So at the end of this lesson, you shall be able to explain the methods to break dormancy. Before that, in the previous video, we have overly seen that the location and cause for dormancy in certain species. Here I have listed few of the crops and their location of inhibitor and also the name of the inhibitors present in that particular crop. Let us see about that. Gossypium species, that is cotton, the inhibitor present the location of inhibitor is pericarp or testa. The name of the inhibitor is ABA. This inhibitor causes the dormancy in this particular cotton. And next one is Coriandrum sativum, that is coriander. The location of inhibitor is pericarp. The inhibitor name of the inhibitor is coumarin. Then next one is Helianthus anus, that is sunflower. The location of inhibitor is pericarp and testa. The inhibitor present here is hydrocyanic acid, that is HC. Next one is Oryza sativa, that is rice. The inhibitor is present in hull. Probably abscisic acid is present here. The next one is Triticum species, that is wheat. The inhibitor present in pericarp and testa. The name of the inhibitor is catechin tannins. The next one is Hordeum vulgare, that is barley. And the next one is Beta vulgare, sugar beet. The next one is Avena sativa, that is oats. Go through this table to know about the location and cause for dormancy in this particular species. The next one we move on to the methods to break dormancy, and there are various methods to break the dormancy. The first one is physical method, the next one is physiological method, and last one is chemical methods. Let us see about the physical methods. The first one is scarification. What do you mean by scarification? Any treatment, it may be physical or chemical, that weakens or softens the seed coat is called as scarification. It is mostly applicable to Malvaceae and Leguminaceae group of seeds. In Leguminaceae group of seeds, the crops has a hardened seed coat, hard seed coat. The scarification may be done by manual, that is physical, or mechanical, acid method, biological or thermal or fire. The first will see manual or physical scarification, that is hot water treatment. Here the seeds are soaked in boiled water for 1 to 5 minutes to 60 to 80 minutes. In some crops like Bengal gram and groundnut, hot water treatment for more than 1 minute is injurious to seed. So careful precaution should be taken while doing a hot water treatment. It is most effective for large leguminous seeds, that is tree crops, particularly Pterocarpus indicus, Azadirachta indica and Terminalia brownie. And the next one is mechanical scarification. This denotes that rubbing or abrasion of seeds against the hot surface. It is done partially to damage the hot seed coat to overcome physical dormancy, to allow imbibition of water over part or the entire seed surface, leading to metabolic process of germination. For example, in case of bitter gourd, sand scarification is employed, for which sand and seeds are mixed in the ratio of 2:1 ratio and rubbed against a hot surface of seed for 5 to 10 minutes. The large quantities in this picture, a nail clipper or hot wire burning is carried out to overcome the seed dormancy. The seeds are rubbed against the rough surface, or using nail or nail clipper or anything. It is and with a knife piercing the needle, or it may be treated with a sand. Large quantities of hot coated seed can be mechanically scarified using concrete mixture with sharp gravel or sand. Special drum lined with abrasive material such as sandpaper, crushed glass, or cement fitted with abrasive disc may be made for this purpose. Here it is a mechanical scarifier. Here through the funnel the seed is poured through the funnel, and a rotating pipe is present here and a concrete cylinder is present. This here the seed rotate in air in the bottom of the cylinder, and the cylinder contains abrasive wall. When the seed hits the abrasive wall, the dormancy or the hot seed coat may broken, and the dormancy can be broken. At the end, the seed is dispersed through the hole in the bottom of the cylinder here. The next one is acid scarification. In this mechanical scarification the precaution should be taken is the seed coat should not be excessively abrated, which cause any damage to the embryo. Therefore, the duration of abrasion should be adapted specific to the crop species. The next one is acid scarification. It is done partially to damage hot seed coat to overcome physical dormancy, to allow imbibition of water over part or the entire seed surface, leading to metabolic process of germination. To scarify the seeds, pour the undiluted acids until all the seeds are wetted. Here, for Malvaceae and Leguminaceae group of seeds, only acid scarification is followed. Use 100 ml of H2SO4, that is undiluted acid of 100 ml of H2SO4 per kg of seeds for 2 to 3 minutes is required. To scarify seeds, pour undiluted acid until all seeds are wetted and stir them for few minutes to 1 hour, and immediately wash them thoroughly in cool running water for 5 to 10 minutes, and to remove all traces of acid. Final washing should be done with slaked lime solution to neutralize the acidity of the seed coat and used water. Then finally shade dry the seeds until wet sowing is preferred. Unless the wet sowing is preferred. And for tree crops, for Malvaceae and Leguminaceae crops, use 100 ml H2SO4 per kg of seed for 2 to 3 minutes. In case of tree crops, the duration of treatment may vary between 10 to 15 minutes to 1 to 3 hours depending upon the kind of seed. And in case of rose seed, treat the seed partially with the acid and then given with the warm stratification. And in this table, we have given the duration of soaking in concentrated sulfuric acid to overcome the seed dormancy. In some leguminous tree seeds like Acacia nilotica, the duration of acid pretreatment is greater than 15 minutes. In case of Albizia lebec, the duration is 40 minutes. 85% accuracy is 85% accuracy is obtained. And in case of Cassia fistula and Delonix regia and so on, kindly go through this table. Delonix regia means flame of the forest. Leucona leucocephala, we know that. Prosopis julifera in Tamil Karuvel Maram, etc. The next one is in case of acid treatment, The precaution should be taken as the only acid should be added to water, not vice versa. There are several advantages in acid tree treatment. That is, it is applicable to many species, not only leguminous crops. The duration of treatment is short as compared to other scarification methods. It is the most effective method of bulk treatment for very hard coated seeds. It requires no special equipment and seeds can be stored for a long period after treatment. The major problems in acid treatment is it implies a serious safety hazard to workers. Seeds are in risk of being damaged by over treatment. It can be difficult to safely dispose of waste acids. The next one is bioscarification. Here it is subjecting the seeds to pre-treatment by making use of animals and microorganisms. It is an important factor in breaking down of seed coat impermeability. It is done to partially damage the hard seed coat to overcome physical dormancy to allow the imbibition of water leading to metabolic process of germination. It is done in the following three ways as animals, insects and microorganisms. First one is animals. The pods of Acacia nilotica are fed to the penned sheep or goat and the seeds are collected from the droppings. The combination of moisture, warmth and the chemical action of digestive juices secreted by the penned sheep or goat softens and renders the hard seed coat as a permeable one. The next one is insects. The termites plays an important role, an important agent for breaking seed dormancy in many parts of the tropics. The termites are fed on the seeds and helps to remove the exocarp. The next one is microorganisms. Here the partial fermenting which causes damage to many seeds can be beneficial in overcoming seed coat dormancy. The microbes decomposes the epicarp and mesocarp over a period of time. The precautions to be taken is it is difficult to make use of these animals or organisms as a controlled pre-treatment of seeds and periodic inspection is essential to ensure that the process is not carried too far. The next one is electrical seed treatment. Here it is a rapid heating of seeds by exposure to radio frequency or that is electrical fields of sufficiently high frequency and intensity done to improve the seed germination by reducing the percentage of hard seeds and distract the insects and parasitic pathogens. The samples of optimum moisture content are exposed to RF electric field usually by placing between a pair of metal plates which are a part of electronic power oscillator. The specimen then becomes a part of electrical circuit and absorbs energy from the electromagnetic field generated between these plates or electrodes. When materials such as seeds are treated, an elevation of temperatures is the most obvious effect. This heating is uniform at all points in a homogeneous sample if the whole sample is subjected to the same field. The rate of heating depends upon the frequency of alternating electric field and the electric intensity or field strength in the material. The RF electric fields improve germination by increasing water absorbing capacity of seed by causing reduction in percentage of hard seeds without reducing the keeping quality. The RF electric field may permit the destruction of insects and parasitic pathogens if their characteristics for RF heating or susceptibility to heat damage are greater than those of their host. The precaution to be taken here is the moisture content is very important in establishing the level at which damage to germination occurs and very likely will influence the beneficial response to some seeds to RF treatment. The next one is fire treatment. It is a method of subjecting dormant seeds to heat generated by fire. The light inflammable litter are spread over the fruits and then ignited. It is done to partially damage the hot seed coat to overcome physical dormancy, and in further it leads to metabolic process of germination. Further, the smoke accompanies fire is also shown to induce germination. The generated heat scorches the pericarp of seed so as to improve the germ seed coat permeability. Scorching, the example here is seeds of Calona vulgaris. Dormancy is broken by fire, and also Enterolobium cyclocarpum Heminia courbaril in India enhances the germination of these seeds by overcoming the seed dormancy. And the precaution here to and the one thing is dry heat caused complete failure of germination in case of Albizia procera and Paraserianthus falcateria in India. The precaution to be taken is it is difficult to adjust the heat of the fire to achieve maximum effect without damaging the embryo of the seed. The next one is soaking in water. It refers to soaking of seeds in cold or hot water for a period ranging from few hours to several days specific to particular crop. It is done partially to damage the hot seed coat to overcome physical dormancy, and it leads to metabolic process of germination. It also helps in leach out the inhibitors causing chemical dormancy. Here, the germination of teak is often poor due to dormancy. So, pre-treatment of the seed is required. That is done by alternate wetting and drying of seed for a week is required to break dormancy before sowing. That means for a week means 6 days is required for soaking is required. That is soaking in water for 6 days is a suitable alternative to alternate soaking and drying in order to overcome physical dormancy in teak, that is Tectona grandis in India. Seeds were kept in a gunny bag and dog the bag in water, preferably in a running stream for 12 hours, then spread in sunlight to dry for 12 hours. This has to be repeated for 1 week, that is 6 to 7 days, and it leads to improve the germination percentage varies from 30 to 50% in moisture teak and 5 to 10% in dry teak, and also it helps to leach out the inhibitors causing chemical dormancy. Example here is coriander, the inhibitor is coumarin, and in case of sunflower the inhibitor is hydrocyanic acid. Soaking in water leach out these inhibitors, and it is helpful in improving germination percentage. The precautions to be taken here is the prescription for hot water treatment with reference to temperature of water and duration of soaking must be followed meticulously if seed coat dormancy is to be removed without killing the seeds through successive heating, and it is not safe for smaller seeds. The next method, so far we have seen the physical methods to overcome dormancy. Now let us move on to the physiological methods to overcome dormancy. The first one is stratification. The method of exposing the imbibed seeds to lower temperature, that is cold stratification, or higher temperature or warm stratification for a period of time. Cold stratification is done to overcome physiological as well as morphological dormancy, while warm stratification helps to overcome physical, mechanical as well as morphological dormancy. First we'll see cold stratification. Here the seeds should be soaked in several times their volume of water before pre-chilling at 3 to 5 degrees Celsius for 48 hours or 41 hours. After soaking, the the water is drained off and the moist seeds are stored at 3 to 5 degrees Celsius for the period appropriate to each species. Storage may be without any medium. that is seeds as such, or the seeds may be mixed with 2 to 4 times of the volume of medium such as moist sand, peat, or a mixture of these two can be used. During the seed treatment period, container shall be opened and mixed with water if surfaces dry off. Example here is Eucalyptus delicatensis and cherry and oil palm seeds are overcome by cold stratification. The next one is how the cold stratification helps in breaking seed dormancy maize. The combination of high moisture and low temperature appears to trigger off changes in cell energy relationship, leading to biochemical activity which transforms complex food substances into simpler forms utilized by the embryo when it renews growth at germination. Low temperature not only favors these biochemical changes but reduces the activity of microorganisms, and there is a risk of overheating and premature germination of the after ripened seeds. Then next we'll move on to warm stratification. Here soak the seeds in several times of their volume of cold water at approximately 3 to 5 degrees Celsius for 48 hours and drain off 48 hours. Drain off the water and mix the seeds with 2 to 4 times of the volume of moist and water retained medium such as sand, sandpeat mixture or vermiculite, and store at a warm temperature of a constant temperature of more than 15, that it may be 20 to 25 degrees Celsius, or alternating temperature of 20 degrees Celsius and 30 degrees Celsius is suitable for many species. Open the containers weekly. Mix seeds if surface shows signs of drying and remoisture with the water spray. The period of treatment may vary from 2 weeks to 16 weeks depending upon the species, and both physically and physiological changes occur in stratified seeds. Partial digestion of the outer layers and final weakening of the inner layers relieves the seeds of physical dormancy. And the precautions to be taken here is the temperature of stratification, moisture contents of seeds, as well as the media should be standardized for better results. Seeds or fruits must be thoroughly dried after treatment. In most cases, it is preferably to accept the safer but slower method of worm treatment. Example here is, for example, paddy. Then, and in case of oil form, it requires a temperature of 40 to 50 degrees Celsius for 2 months to break dormancy. And the next one is temperature treatment. Here the low temperature treatment, plants which grow in temperate or cooler climate requires a period of chilling for to break dormancy. Here apple is an example, can be released by low temperature treatment by storing the seeds at 5 degrees Celsius. And high temperature treatment, normally high temperature treatment are exhibited by early flowering winter annuals, that is bluebell, hyacinthioids, non-scripta. Their seeds are shed in early summer and it does not germinate until they have been exposed to heat during high summer. The next one is alternate temperature treatment. In case of bulrush, that is typha, most it requires alternate temperature for breaking the dormancy, and also most of the plant species which grows in temperature and cool temperature regions requires this type of treatment. And the next one is light and phytochrome. Here the red light of nanometer 630 nanometer and promotes germination, whereas far red light 730 nanometer affects germination. And finally, so far we have seen the physical and physiological methods to break dormancy, and now we'll enter into the chemical methods to break dormancy. And the first one we'll see the growth regulators which are helpful in breaking dormancy are gibberellic acid. It is a light substituting chemical used in wide variety of seeds showing various forms of dormancy. including those with chilling treatment, light treatment, and the need for after ripening and dry storage. It can be used in concentration of 100 to 1000 ppm. It is effective in stimulating germination in large number of species. GA4 and GA7 are even more active, being effective at much lower concentration than GA3. And gibberellic acid is more effective in breaking dormancy of Lonicera, Lonicera maki and Lonicera maroi, etc. Inducement of seed dormancy, inducement of seed germination by gibberellic acid in dormant seed has been attributed because of various reasons, like it increases the growth potential of the embryo, weakens the structure covering the embryo through cell wall hydrolysis, example weakening of endosperm in tomato, and it counteracts the effects of abscisic acid directly or indirectly, and it releases photodormancy by regulating phytochrome, and it promotes germination and thermodormancy through abscisic acid metabolism. What happens when the gibberellic acid is treated with a seed? Let us see. When the water is absorbed by the seed, the gibberellin synthesized by the embryo, and the gibberellin which stimulates the alluran layer to synthesis amylase. The gibberellin stimulates the alluran layer to synthesis amylase, and this amylase hydrolyzes the starch into the maltose, and the maltose is further converted into glucose, and the glucose is transferred again to the embryo for the further growth and development. This is how gibberellin acts on a seed, and it helps to overcome dormancy. The next we'll see that kinetin, and the cytokinins are present in developing seeds and accumulate predominantly in the liquid endosperm. Cytokinin are needed for the promotion of cell division in the embryo, and in many species cytokinin alone break seed dormancy, and cytokinin appear to contribute promotion of dormancy release and subsequent germination by enhancing ethylene biosynthesis. Kinetin will stimulate germination of certain seeds, but it will not completely replace the requirement of red light in lettuce seeds. And the applied cytokinin, that is kinetin, have been reported to overcome high temperature dormancy also in lettuce seed. In case of Ziziphus mauritiana, 100 ppm of kinetin stimulated both the total germination and vigor of seeds, but the effect was generally lower than that of other compounds tested. The next one is ethylene chlorohydrin, which often mimics the effect of ethylene, has also been reported to increase the germination of tree seeds. Ethifone is an ethylene releasing compound, stimulates germination of dormant and non-dormant seeds, although in some cases it may inhibit or no effect on germination. And it is observed that the embryonic axis of non-dormant varieties of peanut actively produce ethylene during germination, whereas those of dormant varieties produce only low level of ethylene when imbibed. So, the treatment of dormant seeds with ethylene induces a high percentage of germination. Similar results were obtained when subterranean clover seed treated with exogenous ethylene stimulate germination of dormant seeds. And also this ethylene or ethifone alleviates the primary dormancy and also light induced dormancy. What happens when the ethylene is involved in dormancy breaking? The primary action of ethylene is it promotes the radical cell expansion in embryonic hypocotyl and increased seed respiration or increased water potential of the seeds. Apart from this, endogenous ethylene also promote germination of dormant seeds by decreasing the sensitivity to endogenous abscisic acid. Ethylene also may counteract the inhibitory effect of abscisic acid on seed germination by interfering with the ABA signaling, and excess of ethylene can bypass the GA requirement. Ethyl can also be used for breaking dormancy of seed coat. The dormancy in cotton seed is due to presence of abscisic acid in the pericarp seed. This we have seen in the first slide, that is the location and causes of seed dormancy in case of Gossypium species. The next one is promoter inhibitor concept. Let us see promoter inhibitor concept. For regulation of germination, the promoter and inhibitors present in the seed should be in a balanced manner. GA and cytokinin are the promoters of germination, whereas the abscisic acid is an inhibitor. It prevents the germination by affecting the RNA synthesis. This and all we have already seen. What is the relationship between these three growth regulators that we'll see in this table? While all these three factors present means it is a non-dormant seed. When GA and cytokinin are present, it is non-dormant because abscisic acid is an inhibitor. It is non-dormant one. And gibberellic acid only present means it is also a non-dormant. When cytokinin only present, it is a dormant seed. When gibberellic acid and ABO are present means it is non-dormant one. When the gibberellic acid is absent and abscisic acid is present, the seeds undergoes dormancy. This table is very important in case of in exam point of view. The next chemical is physicosin. It is a diterpene fluoride, is a toxin produced by the fungus Physicosin amygdalae. It is capable of breaking dormancy and stimulate germination rate of embryo that is isolated from the dormant grains. It induces extracellular acidification, and it had a acidic external pH is able to enhance the GA-induced biological responses such as alluron amylase production. A treatment of physicosin was an effective as like kinetin, ethylene and gibberellic acid in alleviating stress, suggesting that it may affect processes common to both cytokinin and gibberellin. And it was reported that to have no effect on endogenous abscisic acid content but strongly reduces the inhibitory effect of abscisic acid on germination, and it's suggesting that it may directly interfere with ABA signaling transduction. The next one is soaking in chemicals like potassium nitrate. Nitrogenous substances such as nitrate, nitrite, ammonia and thiourea are helpful in breaking seed dormancy. That is, potassium nitrate broke dormancy by decreasing abscisic acid. In seed testing, the recommended concentration by ISTA is 0.2%, that is KNO3 at 0.2%. In case of breaking dormancy, 2% is used for breaking dormancy of light requiring seeds like lettuce. Example here is oats, barley and tomato. The next one is thiourea. The first chemical to break seed dormancy of lettuce was thiourea. Nitrate and thiourea are the possible regulators for seed germination in soils. The thiourea is used for breaking dormancy for both light and chilling requirement seeds. Example here is lettuce. The chemical used here is thiourea at 10 power minus 2 to 10 power minus 3 molar is used. The next chemical is hydrogen peroxide. It is an active oxygen source with relatively long half-life and is able to diffuse into plant tissues. And it have been reported that oxygen could accelerate ABA degradation. Thus, by enhancing the oxygen production, H2O2 decreases ABA content, thereby promote germination. And the example here is it is useful for all crops, and the seed treatment with other growth regulators or chemicals or sulfhydryl compounds like 2-mercaptoethanol or 2,3-dimercaptoethanol can also be used. Commonly, the alcohol compounds also helpful in breaking seed dormancy, and particularly it is seen that ethyl alcohol or ether removed the dormancy in Gossypium species, and ethanol also promoted germination in hypoxia, and other alcohols like butanol, propanol 1, 2-propene 1, these are the good substrate for alcohol dehydrogenase had the same stimulatory effect. This improvement of germination by alcohols, which were good substrate for alcohol dehydrogenase, always associated with increase in oxygen uptake by caryopsis, whereas alcohols which were not ADH substrated did not enhance respiration. So, alcohols are metabolically involved in stimulation of germination as a respiratory substrate, and it is involved through the activation of glycolysis and Krebs cycle. And also the plant products like strigol, that is a root exudation taken from a striga parasitic plant, can also be used for breaking seed dormancy. Thank you friends. Thank you for watching this video, and if in this video the dormancy topic is over, next we will see with the interesting topic. Thank you for watching. Kindly watch, like, share and comment on this video. Thank you.